All right. I was going to ask if the mic was on, but I can tell it is. All right. Good morning, everyone. Thank you so much for coming here, especially considering how busy this meeting is and how many competing events there are. Here we are at the very first international student debates that have been held at ICE. I wanted to start by thanking our organizers. We have 2 student affairs committees this year because of the nature of ICE needing long-term planning. So we have members of both the ICE Student Affairs Committee helping with this, including myself as co-chair of the ICE SAC, Tamara Real, and Richard Haypenny, who could not join us today. From the ESA side, we have the current co-chair of the ESA SAC, Kendall Dye. We also have Alex Whitener, who's up in front helping us with timekeeping, and then JD Gantz also assisted. So we have a social media component to this, just like a lot of the other events here at ICE. Please live tweet or post about the student debates using the debates hashtag and the ICE hashtag that you see on the screen. This gives you a chance to win a gift card, and we'll enter you in a raffle for that card. So we have a few topics that will be presented here today. Our very first debate is what will be the single best policy for improving health of Apis mellifera if adopted worldwide. We already have those 2 teams on stage, University of Delaware and Midgrass Prairie Regional Team, which is interesting since it combines 2 different universities. The second topic after the first will be what is the single best strategy for decreasing dengue fever virus incidence worldwide, and that will be done by North Carolina State University and Purdue University. And then the final topic will be about choosing the most important invasive arthropod species internationally that needs to be controlled. The 2 teams debating will be Auburn University and the International Center of Insect Physiology and Ecology. I wanted to make special note about that second team. For various reasons, including funding and visas, only 1 team member could make it, and we had very kindly a second team member from Australia join him in the true nature of ICE. So when we get to that debate, I'd appreciate if everybody was very attentive and congratulatory of that team for pulling together. So thank you so much, and I will let our moderator, Kendall Dye, take over from here.
Thanks, Rebecca. So I'm going to detail the debate structure just for a few minutes so everyone is on board, especially the audience. So we will start off with an unbiased introduction. This will be from a student who is not affiliated with any of the universities who are debating for each of the debates. They will introduce the topic. Then the first team will give an introduction on their side of the debate. This will be a 7-minute introduction. And then there will be a cross-examination period for 3 minutes. The cross-examination period, it's very important to note, is a back-and-forth question and answering session. When we go into the Team 2 introduction, they will also introduce their debate side, and then there will be a second cross-examination session. When we enter into the rebuttals, these will only be 3 minutes, and these are not question and answers. These are only questions from one team. For example, when Team 2 enters into its rebuttal period, it can ask questions to Team 1, but Team 1 cannot answer until their rebuttal session. So that's very important to remember. After 2 rounds of rebuttals on each side, there will be a question session from the judges and the audience. The judges will ask their questions first, and this will be a 10-minute question and answer session. And if there's time left after the judges have finished their questions, then we will have questions from the audience. So we have a floor microphone available. If an audience member does have a question and there is time, the floor mic is right here, down there at the front. And so if you could line up at the microphone, that would be great. And then we'll get your questions asked, and then the teams can answer. And also, 1 person, 1 question, please. The winners of the student debates will be announced at the student awards ceremony, and this is going to be Friday, so tomorrow afternoon from 12:30 to 2:30 p.m. So of course, be sure to be there to support the winning debates teams and also all the other teams who have debated today, and to find out, of course, who won. And that will be here in the Chapin Theater. So moving on even to next year, if anyone is interested in debating next year, it would be great if you could contact the Student Debates organizing committee, let them know you're interested in forming a team, and start getting ready for next year, and that will be in Denver 2017. So of course we need to thank everyone. There is a lot of work that goes into organizing, debating, and judging these debates. So we'll thank again our co-organizers that Rebecca introduced. We have some judges that we need to thank. Sydney Crawley, and we did have some judges missing, but we are working with that, and we're going to fill those in with some people who are qualified, including Rebecca. So also we need to thank our ESA staff, Debbie Sutton and Rosina Romano, who have been crucial in helping us. Our advisor, Ann Nielsen, of course, the teams and their advisors, and all of our introduction speakers, and of course you, the audience. And so with that, we'll get into it. Okay, so if we could have our first unbiased introduction speaker. And also for the teams, there's a timer, of course, you can see it. So I will be cutting you off if it's time.
Okay. Thanks everyone for joining us for this debate about what would be the single best policy for improving the health of Apis mellifera if adopted worldwide. I'd like to spend the first couple of slides, actually in a little tangential fashion, trying to convince you really that coming to really firm, conclusive findings about honeybees at the colony level is kind of a difficult thing. So I'll do that with a couple of situations, both past and present, that kind of illustrate this. And the first is actually one that I read about recently in a paper by ESA President May Berenbaum in the Journal of Agricultural and Food Chemistry. And it's about the initial debates surrounding the effects of arsenical insecticides on honeybees in the early 1890s. And so actually, the American Association of Entomologists ended up forming a task force on this issue. And after several years of investigation, the final final findings were published by a guy named Lintner, and ultimately they were relatively inconclusive. And so there were many reasons why they were inconclusive, and some of them were as simple as poor bee husbandry. So, keeping a honeybee colony alive in a highly controlled experimental setting is not the easiest thing in the world. So, for example, in one experiment, the experimenter had tried to have a whole colony of honeybees under a stone fruit tree, and he put netting around it and sprayed the tree with insecticides. And the bees died, of course, but he couldn't conclude whether or not it was the insecticide or whether it's just the bees couldn't get out of the netting, because you can't confine them in an environment like that. So things like bee husbandry, the lack of basic toxicological parameters such as the median lethal dose, as well as apparent lack of the concept of an experimental control really hampered research at this time. Of course, bee research and research in many fields has moved forward significantly over to modern day. But even now, we have really considerable, intense debates about how certain factors, or what factors, are causing decline or poor health in honeybees, rather. So one thing we can think about is the recent colony collapse disorder phenomena occurring in late 2006, which really sparked an intense media frenzy, and the best bee researchers from all around the world came together to try to get to the root of this problem, and ultimately fell short. They could not find the cause or the solution. Luckily, the phenomena quickly dissipated over the following several years, but you can see, even when we've got the best scientists of the time on the issue, it can be difficult to figure out what's going on with honeybees. And so I want to bring up a table, again, from this paper by Dr. May Berenbaum. And it's sort of just about the systemic difficulties of doing experimentation with honeybees at the colony level. And so, if you think about comparing honeybees to most insect systems, honeybees are different in many very significant ways. For example, the unit of replication for most insects is the individual, an individual fruit fly, for example. But a honeybee colony is a superorganism. It's thousands of individuals. The size of that colony varies across time. The genetic makeup of the colony is constantly changing. The lifespan of the individuals composing the colony is varies seasonally and by caste. The food that's brought into the hive is rarely consumed by the individual that collected it. So there are these structural factors that make working with honeybee colonies much more variable than other types of insects when we're talking about research. So with that being said, we're obviously not talking about the difficulties of honeybee research or arsenical insecticides. We're talking about which factors are most negatively affecting bee health. And I think if you talk to most bee researchers, you'll find that there are really 4 key factors: parasites, poor forage, pesticides, and pathogens in no particular order. Obviously today, these student teams have chosen two topics, including poor forage and pesticides. I think that's really interesting. I think actually, in my opinion, if you talk to many bee researchers, you'll find that these 2 topics of choice are very interesting because they can sort of be, I think you make a strong argument that they're rooted in one central change that we've been, that landscapes have been undergoing in recent times. When we think about increased agricultural development and increased urbanization, which has really resulted in changes in the chemical inputs that landscapes receive, as well as the structure of landscapes, and ultimately is changing the forage that bees are encountering. So given that those massive changes in landscape composition, you can see how you can make a strong argument that both of these factors, poor forage and pesticides, can be contributing to poor honeybee colony health. So with that being said, I think it's going to be a really interesting debate. There's a lot of material here, and I'm really interested to see what these teams have brought to the table here. So with that, I'll hand it over.
Thank you, Rodney. So we can now move into our Team 1 introduction.
Okay, thank you very much for that introduction. The Midgrass Prairie Regional Team, comprised of students from both Kansas State University and South Dakota State University, would like to welcome everyone this morning to the debate, and we feel honored to sit across from the fine students of Delaware and wish them the best of luck today.
Shouldn't we have a presentation?
Yes.
Okay. This one?
That's it. All right, thank you very much. To put colony collapse disorder into perspective and to relate it to the human species, I would like to read a quote from Goulson in 2015. Unfortunately, the public debate on bee health has often become polarized with claims, for example, varroa or neonicotinoid insecticides are the sole or primary cause of bee declines or honeybee losses. If a middle-aged man who is overweight, does little exercise, smokes and drinks heavily, were to die of a heart attack, we would not be surprised, and we might not spend too long arguing over which single risk factor was most important in bringing about his untimely demise. Similarly, wild bee declines and honeybee colony losses are clearly due to multiple interacting and sometimes synergenic factors. And the combination of factors involved no doubt varies in time and space. End quote. To properly understand interacting synergistic factors between neonicotinoids and other factors responsible for honeybee decline, we must first understand neonicotinoids and their effects on honeybee health. The effects of neonicotinoid insecticides on honeybees necessitates that use of these products should be restricted. Mounting research is revealing that the class of neurotoxic acetylcholine inhibiting insecticides produces a growing number of sublethal effects on honeybee health. These effects include decreased foraging success, impaired navigation, suppressed developmental hypopharyngeal glands, inhibition of abdominal ventilation movement, negative impacts on the honeybee immune system, and hive reproductive capacity. Moreover, effects of neonicotinoid insecticides aggravate other stressors on honeybees. For example, the negative effects of varroa mite and nosema infestations and infections with deformed wing and black queen cell viruses are compounded by interactions with neonicotinoids. Management decisions must weigh the agronomic value against potential environmental harm. Scant evidence supports the agronomic benefit of neonicotinoid seed treatments. Substantial consensus among scientific studies suggests that pests are not reduced and yields are not improved in a number of seed-treated row crops relative to untreated fields or fields treated using IPM action thresholds. Despite the lack of evidence for economic benefit, the use of neonicotinoids in agriculture remains widespread. At least 42 million hectares, or 104 million acres, of 3 crops, maize, cotton, and soybeans, are planted with neonicotinoid seed treatments every year. Maize, 79 to 100% of the crop, cotton, 52 to 77% of the crop, and soybeans, 33 to 44% of the crop. For example, more than 1.2 million pounds of thiamethoxam alone was used in the United States in 2013. These neonicotinoids can persist in the environment for multiple years, and they move out of cropland into neighboring habitats, where they affect aquatic systems and untreated vegetation that bees need for forage. It appears that this environmental contamination is the result of at least leaching and wind dispersal, which includes planter dust. This environmental contamination of bee forage by neonicotinoids increases the exposure of bees to these toxins. Honeybees are unable to distinguish between neonicotinoid-laden nectar and nectar from untreated plants, and even consume greater quantities of the treated nectar. Field collections of pollen, bee bread, and nectar are contaminated with biologically meaningful levels of neonicotinoid toxins, even in entirely untreated habitats. These field exposure levels were correlated with reductions in honeybee health. Thus, honeybees are unable to avoid consuming hazardous levels of neonicotinoids in agricultural regions of the country where they spend most of their time. The ubiquity of neonicotinoids has resulted in an environment nearly void of uncontaminated foraging habitat. The need for quality forage is undoubtedly crucial to the health and survivorship of honeybees. However, this scenario is extremely challenging and entirely impossible whilst neonicotinoids are used at the current rate and with no restrictions. Given these arguments, increasing bee forage can only benefit pollinators in the presence of restricted use of pesticides. For this reason, restricting the use of neonicotinoids should be prioritized over increasing bee habitat in the environment. And here are our list of references. Thank you very much.
Thank you very much. And we will have a 3-minute cross-examination period once that gets loaded. Okay. Team 2, you can cross-examine Team 1.
We have no further questions.
Okay. Team 2 can now give their introduction presentation.
So, University of Delaware, our team believes that the single best policy for improving the health of Apis mellifera worldwide is increasing their access to varied, high-quality forage. And I'd like to begin just by reviewing some basic facts about honeybees, which I'm sure everyone's already familiar with. And some of this will be recap based on the wonderful comprehensive introduction we had from our unbiased introductor. But anyways, we believe that honeybees are an extremely important species to humans because they are efficient generalist pollinators, and in fact, they are the single most important pollinator for most of our crop monocultures today. An estimated 35% of the human diet benefits from pollination, and pollination services are worth an estimated $215 billion globally each year. Although the number of honeybee colonies is on the rise worldwide, there has been a marked and prolonged decrease in colony numbers in both North America and Europe, and currently demand for honeybee pollination services vastly exceeds availability, and this trend is likely to continue in the foreseeable future. Honeybees are facing an onslaught of stressors whose effects may well be synergistic rather than simply additive, combining to debilitate bee health at the individual, colony, and metapopulation scales. These threats include parasites, such as Nosema fungus and Varroa destructor mites, the latter of which vector deadly pathogens such as deformed wing virus and acute bee paralysis virus. Additionally, bees suffer negative effects from exposure to pesticides, namely those in the neonicotinoid and the organophosphate classes. The ever-looming threat of climate change poses unpredictable challenges as it induces range shifts in the plant species that honeybees depend on for nectar and pollen nutrition, and also subjects honeybees to extreme temperatures and extreme weather events. Most importantly, honeybees are subjected to starvation and malnourishment due to reduction in the quantity and the quality of available forage, rendering them far more vulnerable to the threats described here. As the human population has doubled over the past half-century, we have observed dramatic shifts in land use practices, namely a transition from natural and semi-natural open areas to more urbanized landscapes, which are marked by increased development and a concurrent decline in pasture. Small farms growing a diversity of crops have given way to large, intensively managed crop monocultures. The use of synthetic fertilizers has led to a decline in the practice of crop rotation, as well as the use of cover crops. And the use of herbicides eliminates weeds that would provide additional varied sources of nutrition for honeybees. These factors combine to create effective food deserts for pollinators. Landscapes marked by short, synchronized flowering periods that do not meet the bees' nutritional needs over the long foraging season. The lack of availability of floral resources in agroecosystems has resulted in malnourished, unsustainable honeybee colonies, which is widely considered a leading cause of colony losses here in the United States. A single plant species rarely provides the full suite of nutrients that honeybees require, so access to varied sources of nectar and pollen are crucial to combat starvation and malnutrition and to promote proper growth and development. Honeybees which are deficient in one or more of the 10 essential amino acids are limited in their ability to rear brood. They also experience impaired development of hypopharyngeal glands, ovaries, and flight muscles, and may suffer an overall shortened lifespan. Bees under stress from factors such as disease have heightened energetic demands and increased need for bountiful floral resources to overcome infection. Bees whose nutritional needs are not adequately met are more likely to succumb to environmental stressors such as climate change and toxic pesticide residues. Due to concerns about food security, there's always going to be a demand for effective pesticides, It is unrealistic to expect full international compliance with restrictions on pesticides, which are valued for their ability to protect crops and increase yield, thereby reducing the total acreage required for crop production. Additionally, restricting usage of one class of pesticides may have short-term benefits for pollinators, but it opens the door for other potentially even more harmful chemical applications to fill that void. What we really need is an alternative, long-term ecological approach to honeybee conservation. Making our policy a reality is as simple as following these 3 general guidelines. We want to incentivize plantings of wildflower refugia in agricultural areas. These would include flower-rich field margins, hedgerows, and biodiverse meadows. We also want to provide a diversity of flowering plants throughout the foraging period and improve habitat connectivity to facilitate bee mobility between habitat patches. This would reduce foraging distances and allow for increased pollinator population growth. In conclusion, unfortunately, these problems are so complex that there is no single silver bullet approach to protecting honeybees, because they are facing an onslaught from multiple interacting stressors. But we do know that improved access to varied high-quality forage improves their nutrition, immune function, and overall health, thereby allowing us to mitigate multiple stressors simultaneously with a single policy. Alternative strategies, such as restricting pesticide use, doubtless do have potential to improve bee health, but their application is complicated by conflicts with other agricultural objectives, namely pest control. Reducing or restricting use of neonicotinoids could have severe implications for economic and food security. By increasing honeybees' access to varied high-quality forage, we bolster their ability to withstand the complex and synergistic pressures of their ever-changing environment. Thank you.
Thank you, Delaware. Team 1 can now cross-examine Team 2 for 3 minutes.
Okay. In slides number 4 and on your conclusion slide, you mentioned the importance of varied forage. And in one of your citations, D. Pascal et al. 2013, they found that bees which consumed a diversity of pollens never outperformed bees which ate a monoform diet of pollen with the highest nutritional value of those within the mixture. With this in mind, how do you further support your statement of increasing varied plant diversity for the benefit to honeybees?
Loper and Burdell in 1980 showed that honeybees need a variety of different pollens to meet all of the nutritional requirements, primarily amino acids. And with the prevalence of these large monocultures of crops, we're seeing less and less diversity in the landscape. And so in a lot of these cases, we have only very limited floral resources for the bees to use. So while you may have a threshold at some point where you're meeting all these requirements, having a varied diet is going to give the bees the best chance of meeting all of those nutritional requirements.
An additional reference would be Rulston and Kane, 2000. And this paper shows that honeybees that were fed only on dandelion pollen died sooner than those fed on others, those fed on a variety of pollens.
I had a question. So you brought up the Looper study discussing how landscape diversity was increasing the diversity. So in another one of your papers, the Smart et al., I was hoping you could clarify, they mentioned in it that greater landscape diversity and amount of uncultured forage surrounding an apiary did not equate to greater diversity of collected pollen in those apiaries. So I was wondering if you could clarify how the bees are utilizing that diverse landscape around them to increase their pollen.
So in the SMART paper, they did find that apiaries in areas with increased uncultivated forage had better overall winter survival. So having these more diverse foraging areas.
I'm sorry, that was the 3-minute period. Now we can move into Team 2's first 3-minute rebuttal period. And just 1 second. Okay. Team 2, first rebuttal.
Sure. The Mullen et al. Paper in 2010, this was one of our references. They did a broad spectrum, or a broad survey of different pesticides and they found residues of 121 different ones, of which only 6 were neonicotinoids. So it's pretty clear that honeybees are facing an affront by a varied cocktail of agrochemical compounds. In fact, there were 16 pyrethroids, 16 organophosphates, 17 herbicides, 30 fungicides, and so on. And pyrethroids were cited as the most frequent class found and are also known to be associated with bee kills. So we are just wondering what led you to focus specifically on neonicotinoids, and also if neonicotinoids were to be restricted, what would prevent farmers and other users from simply filling that void with other chemicals?
Yeah, Rodney brought up in the introduction that we first became concerned about pesticides and honeybee health in the 1890s. And there's been a lot of attention on neonicotinoids and a lot of research into it. Clearly, they're having an effect on honeybee health. We're not denying that. But there's also the concern of pest control, whether it's neonics are actually helping pest control or if there's a perceived effect on yield. There's still going to be a demand for pest control with some type of chemical after we remove neonics. And so banning neonics is just going to create a market for a new chemical. So if we want to look at this in the long term, we need to think about what are we going to do to protect honeybees against all of these different stressors? How are we going to keep them healthy as we figure out the best way to control these chemicals to deal with diseases and pathogens?
Yeah, I'd like to follow up on that by saying the effects of neonics, like you said in your introduction, vary in time and space. Certainly in the midgrass area and regions where farming is rather ubiquitous, certainly drift from seed application neonicotinoids does cause a big issue to bee health in those areas. But in areas where these practices aren't so large, for instance, near cities like where we come from in Newark, Delaware, farming is not a big undertaking, and really the issue facing honeybee health in these areas is decline of foraging areas.
Thank you. And Team 1, you can enter your first rebuttal period of 3 minutes.
Thank you for your questions, Delaware. We do recognize that in the Fraser paper, they do find a variety of chemicals within bee products. I would also like to respond to that by bringing up one of your citations, Gill et al. 2012, in which case they showed that there were negative effects on foraging success of bees exposed to imidacloprid in comparison to unexposed bees. But when bees were exposed to imidacloprid as well as cyhalothrin, the effects were significantly more severe even than the single insecticide exposure. And we believe that this demonstrates that by removing one chemical, the bees will respond and perform better than if they are to be exposed to a multitude of insecticides.
You've referenced in your PowerPoint a couple times food shortage. You can talk about that stuff, but 2 references we cited, Seagraves and Lundgren, Bredesen and Lundgren, showed no significant bump in yield at all with or without neonics, and no increased pest pressure. So how, I'm just curious, the thought process in a ban of neonics causing a potential food shortage. If yields aren't affected, pests aren't affected, where is that going to come from?
Yeah. Also, just to answer, I guess, get at your question of if we ban neonics, won't farmers replace them with a potentially more harmful insecticide? So yes, we do recognize that banning neonics could lead farmers to replacing them with more harmful pesticides. However, this is a big assumption, and there's no guarantee that the farmers will necessarily switch to insecticides. They could reduce their pest problems with other IPM methods or programs. Furthermore, Douglas and Tooker from 2015 show that neonic use in the U.S. is primarily applied as seed treatments through 3 crops, and the work by Bredesen and Lundgren, as well as Seagraves and Lundgren, demonstrated that neonic treatment is not adding pest control to many of these crops, which highlights the potential for no adverse effects from banning neonicotinoids under a large portion of how they are applied within the U.S., which would lead the farmers to not really needing to replace them as much.
That concludes our responses.
Okay, thank you. We're working on the clock.
Yeah.
We had,
No. Yeah.
oh, did we
Yeah.
run out of time?
Yeah.
Oh,
You had like 15 seconds left.
okay.
Sorry.
No, that's fine.
Next.
Okay. Now Team 2 can go into their second rebuttal of 3 minutes.
One of your references is the European Academy's Scientific Advisory Council 2015. Although this tome did focus on neonicotinoids as a driving stressor in honeybee populations, they also made the point in their introduction that the Commission's 2013 decision to ban neonicotinoids for 2 years does not imply that other factors are not also important potential contributing factors to ecosystem service declines.
The Douglas and Tooker and some of your other references are specific to the United States, but if we're looking on a global scale. And so in the Higgs 2010 paper, they did a large-scale survey of apiaries in Spain. This is one of your references. They looked at pathogens and chemicals associated with honeybee colony depopulation and found that none of the 9 pesticides they looked at were related to depopulation, but Nosema cerana and Varroa destructor were more closely related. Also, the German honeybee monitoring program found no link between pesticides and colony losses on the colony level. So while there are these definitely individual effects on individual bee health and probably some effects on colony level, it seems that honeybees, in some cases, and probably when they have enough forage resources, are able to compensate for some of these. So having a diverse pollen diet can protect them against Nosema infection. That is, I lost my reference here.
Well, just finishing that up. The Woodcock et al. 2016 paper, in their conclusion, they said, while the evidence presented here shows that neonicotinoids are a contributing factor to reduce population persistence, it is unlikely that the effects of neonicotinoids would act in isolation of other environmental pressures.
D. Pascal 2013 was the paper that showed that a diverse, high-quality diet supports hyperpharyngene gland development and immune system health and can protect against Nosema.
Thank you, Team 2. And now Team 1 can go into their second 3-minute rebuttal period.
In reference to the mites, one of your citations, Van Engelsdorp, 2010, talked about the level of infestation in varroa mites causing colony damage has decreasing over time. In the 1980s in Europe, colonies could have several thousand mites without any real dramatic symptoms in honeybee hives, but today, a fall infestation of about 10%, and it cited 1,000 mites in a colony of 10,000 bees, was considered to be a critical threshold for any survival. So, yeah.
As we highlighted earlier, there's usually, in our presentation, there's an additive effect with the neonics. So neonicotinoids alone, like we see when they're applied in Australia, which doesn't have the varroa mite, usually don't have as much of a negative impact on mortality. However, when they're combined with a lot of these other known honeybee killers like the varroa mite, nosema, you see a bigger impact with neonics working with a lot of these other killers additively or synergistically with them. And those other, the varroa mites is throughout much of the Europe, United States, North America in
general. You referenced the Hedges paper, talking about how they reached the conclusion that nosema was the main cause of unhealthy populations. And just wondering how you address the fact that of the samples they tested, only 3 had fipronil, which is an organophosphate, and none of the samples had any neonicotinoids or imidacloprid that they tested in that study. So reaching the conclusion that nosema was the main effect when other pesticides weren't tested or were only tested with a small sample size.
We also had another kind of clarification point here. So in their Smart et al. Paper, where they did some model indications, and they found that if beekeepers sought to achieve 80% survival based on uncultivated forage land alone, it would require approximately 32 hectares, that's approximately 64 football fields, of uncultivated forage per beehive. So we're just wondering, with the increasing world demand, a lot of this land use, especially for food production systems, how are you planning on increasing an area that large to increase honeybee health worldwide?
And then finally, of course, it's also good, according to your reference, Rolston and Kane 2000, that improving high quality forage, you should keep in mind that some pollens could also be toxic to honeybees.
Thank you. After those rebuttals, then we can now have a 10-minute question period, and we would like the judges to ask any questions that they have first. If audience members have questions, please line up at the microphone, which is on the left-hand side to you, out down at the stage. And we can also have someone, yeah. Okay, so Rebecca, you have a question?
On this one, there we go. So a lot of your references are coming from a lot of US researchers, and I know you did bring in some international references, but I'd like both teams to just take a quick minute to talk about how both of your policies would affect in the international community going beyond the United States, and also very briefly mention how maybe the other team's policy would affect the US, outside the US. Thanks.
Go ahead.
We think one of the strengths of our policy is that it's not a top-down approach, it's a bottom-up grassroots approach that anyone can take the initiative to do. Anyone can, on their own land, provide flower margins or hedgerows. So you don't need to wait for the delay of international cooperation, coming. It can be begun at any time. And so it could be done anywhere in the world at any level without, it doesn't need to be a governmental mandate coming down.
Certainly. So large part of our policy was based on the European Union mandate from 2013, kind of restricting the use in Europe, which kind of demonstrates that this can be done across a continent with several different countries. So it has been implemented there, and it kind of demonstrates it can be done across the globe a little bit, working with multiple countries.
Okay, does our other judge have a question?
Okay, I have one question for each team. So we'll start with the natural forage. So I don't disagree with the potential benefits that you've outlined for having these additional foraging sites, but obviously we've moved to monocultures for a reason. And I was wondering what you think the challenges would be to adding more natural foraging sites, and with all these benefits, why did we not see more of these in the field? So that's my question for you. It essentially address the challenges that you see with making more of these natural foraging sites. The neonic group. I agree with you that they didn't cite a lot of literature regarding this gap. If you were to ban neonicotinoids, that there's automatically going to be another chemical that comes out into the field. I agree that there was some lack of evidence for that statement. But what do you think? Do you think if you were to ban neonicotinoids, we do rely a lot on chemicals, what do you think the fallback would be at that time? Just your opinion and some projections. So those are my two questions for each team.
Okay, Delaware, you can start.
So in regards to some concerns as to whether or not adding, or some of the limitations that might occur from adding these kind of high-quality forage areas. In the United States, there's been issues of trying to incorporate native refugia in these farmlands, particularly in the Midwest area, mostly because most of the farmlands that had originally done this through the Conservation Reserve Program, or CRP lands, have been slowly diminishing their returns to the farmers in the past few years, actually in the past few decades, as there's been an interest in increasing biofuel market. So people have been moving their croplands from receiving these government subsidies in CRP lands to something more along the lines of providing corn for bioethanol. As such, you had a kind of removal from these areas of high-yielding, high-quality crop forage or flower forage for pollinators to something that's much more for economic purposes. So there's definitely a money incentive right there that kind of makes this a difficult issue. But the thing that we focus on with, as a main issue with this is that it's been stated several times that land intensification, land modification is increasing. Both the amount of land that's being converted over to what we refer to as food deserts, where there is no available food forage and no available native plants to be used, no available nectaries to be used for these kind of purposes. But this can be done on small scales too. These don't necessarily need large swaths of available forage, but what they depend on is a good variety, a good diversity. In the East Coast, as an example, we have a variety of goldenrod plants, for example, have a decent amount of nectar, not just for honeybees, but for other pollinators as well, that would provide that opportunity for them. And they exist in small patches on farmlands, on sides of roads, on disturbed habitats that would be able to provide the amount of nectar and the amount of food resources that those pollinators would need to survive. So this can be done at a smaller scale, not just simply at a large scale, and those can outweigh the economic cost, so to speak, if someone still wants to convert their CRP land over to those high-yielding, more economically feasible methods.
Thank you. And now, Midgrass Prairie.
Yeah, it would obviously be very difficult. Any insecticide that's used in such large quantities and such a large scale would be difficult, and there would be some growing pains. But I think once you start seeing that remove, that huge stress on honeybees taken off, and being able to hear the advocacy from the beekeepers, who the bees' ability to pollinate in their honey is their crop, just like any other farmer, and able to see such a huge bump in that, and being able to see what kind of services can come from their increased pollination, how that increased pollination can help the surrounding cropland, I think that would ease the going on with that process of getting away from a pesticide-dominated or neonicotinoid-dominated landscape in favor of some more natural pest control and usage?
Thank you for the question. I've got a couple of points as well. If we were to ban neonicotinoids, we realize that every day scientists are creating new technologies, and they are often more specific and applied in ways that reduce the impacts to non-targeted insects. So that is one thing that even the turnover to the next technology might potentially reduce the hazards to bees and other beneficial insects. And also, on an even better note, farmers everywhere are on their own adopting non-chemical integrated pest management schemes. So if there were to be a ban of neonicotinoids, it might push producers to themselves look at integrated pest management options, which might also increase uncultivated areas with forage in them to be uncontaminated.
Thank you. I think we do have time for an audience question. Is there? Okay.
Sorry, I tried to cut the judges before. A two-part question kind of for both teams. So going back to your prompt of what is the best policy, policies are really hard to write and come up with. So for this team over here, just the strategy of changing the ecosystem is a hard, complex problem itself. So creating a policy to be able to regulate that could also be very complex. So just your response to that. And then for this team over here, while neonicotinoids, there has been evidence that they have been harming the bee population, they are comparatively non-toxic to a lot of other synthetic chemicals that are out on the market right now. So a lot of farmers do like them because, again, they're generally not as toxic in the environment. So your response to that? Thanks.
To both teams, please keep in mind that the other team has a question, so kind of make it as quick as possible. We'll start with midgrass on this one.
Okay, again, it's good to keep in mind that farmers are also on their own in adapting integrated pest management. So there are other non-chemical methods to control these pests. As well, it's important to keep in mind that neonics are very widespread and they have the ability to leach into neighboring foliage and cultivars.
And very quickly, I would also like to note that you had mentioned the low toxicity to non-arthropods. There are plenty of studies showing the multi-trophic level interactions with reduced forage for higher trophic levels in habitats with fewer arthropods.
The Pollinator Health Task Force of the United States has already done many millions of acres at this point on restoration land, especially through the I-35 initiative. One of the ways they've done this is actually creating native bee habitat or pollinator habitat from one part of the U.S. to the other through the Interstate 35, so it's possible.
Wonderful. Thank you guys very much. And so that concludes the first debate. So we can thank both of our teams. Thank you. Okay, we are going to have a 10-minute break period. So everyone come back in 10 minutes. Okay. Good morning, everyone, and thank you for coming to the second debate of the morning. So we have NCSU and we have Purdue University here to debate on what is the single best way to decrease dengue incidence worldwide. So if we could have our unbiased introduction first. Okay.
Okay, thank you. Good morning, my name is Ashley Yates, and I'm a graduate student at The Ohio State University. Today I will be introducing the unbiased introduction for the debate topic, what is the single best strategy for decreasing dengue fever virus incidence worldwide? The global range of dengue has expanded since the 1940s, and this expansion can be attributed to mainly three main factors, including global travel, increased urbanization, and a lack of consistent vector control. One of the main vectors of dengue is the mosquito Aedes aegypti. And this insect also vectors viruses that can cause yellow fever, chikungunya, as well as Zika viruses. And due to this expansion, dengue has been classified as one of the most important mosquito viral diseases by the World Health Organization in 2012. This illustrates the need for successful vector control. Dengue virus is caused by one of four closely related single-stranded RNA viruses, and symptoms of dengue include fever and joint pain, as well as a combination of other symptoms, such as vomiting and rash. While the majority of patients that contract dengue do not progress to severe dengue, those that do will experience symptoms including loss of plasma and going into shock, as well as hemorrhaging and organ damage. However, with proper treatment in a hospital, severe dengue can be fatal in less than 1% of patients. It's estimated that 390 million people are infected with dengue annually, and most of these will exhibit mild symptoms. Aedes aegypti has an urban life cycle, as its eggs are oviposited into containers that are collected with rainwater outside near homes. And the adult females do require a blood meal for this oviposition. And maybe most importantly, these insects prefer to feed on humans rather than other animals, as well as live inside of our homes. Traditional methods for Aedes aegypti control have included removing the water from these containers that collects outside of homes in order to remove the oviposition site. However, this does require long-term participation from entire communities for this to be truly effective. Insecticides have also been widely used for Aedes aegypti control. However, there have been documented cases of resistance development in this insect, as well as overuse of insecticides can cause harmful negative environmental effects. Therefore, new methods of vector control are needed, with the goal of vector control being to support our long-term goal, which is to slow the transmission of dengue. And considerations for these novel methods include targeting the adult populations, as the adult are the vectors of dengue, and specifically targeting the mating mechanism, as this helps to ensure that the novel control methods will be species-specific. These control methods typically address one of two goals: to suppress the population, where a novel trait is introduced into the population to cause lethality, or to replace the population, where a trait is introduced that disrupts the ability of the mosquito to transmit dengue. The two control methods that we'll hear about today are genetically engineering Aedes aegypti with a lethal gene and biological control using Wolbachia. Genetic engineering of insects with lethal genes is conceptually similar to the sterile insect technique, except for instead of using radiations, lethal mutations are generated using techniques in molecular biology. But it's similar in that the progeny of these mosquitoes will then express a lethal gene. And some of these techniques can be specifically designed to target female progeny. The other method, to transform with a bacterial species of Wolbachia, and Wolbachia is a maternally transmitted bacterium that's found in many insect species but is naturally absent from Aedes aegypti. An introduction with Wolbachia can prevent viable offspring by cytoplasmic incompatibility. So for example, if an infected male is mated with an uninfected female, these offspring will not be viable. And there are some strains of Wolbachia that can actually make Aedes aegypti less susceptible to dengue viral infection. Thank you for your attention, and good luck today.
Thank you. We will now have our first introduction from Team 1.
As Ashley just explained, dengue is a serious global problem, as are the mosquitoes that spread it. There are 4 dengue serotypes, with a 5th one putatively identified. The serotypes are distinct, with only 70% genetic similarity between them. How dengue ultimately impacts humans depends on a number of genetic, environmental, and host factors, many of which remain uncertain today. While people can be asymptomatic with a primary infection, a secondary infection with a different serotype can lead to severe and possibly fatal cases of hemorrhagic fever. Current methods to decrease mosquito populations have not decreased the incidence of dengue fever. We need new methods, ones that eliminate mosquitoes and the viruses they carry, not ones that merely reduce the chance of transmission. Releasing Aedes aegypti mosquitoes that are modified using lethal gene technology is the best and most promising approach for stemming the dengue epidemic tide. Lethal gene technology kills mosquito larvae before they become adults. In essence, this creates a dead mosquito, and a dead mosquito cannot transmit dengue. We will present 4 main arguments why lethal genes are a superior technology. They're more targeted, they're reversible, they're versatile, and they're more responsible. Genetic modification is a frequently used, decades-old technique. Scientists insert a small number of lethal genes into with well-characterized mechanisms into the mosquito. Promoters are chosen to express these killer genes in a specific and desired spatial or temporal pattern. Lethal gene technology has the advantage of being reversible. If any negative effects are observed, lethal genes will disappear from the environment soon after releases are stopped. A large library of naturally occurring lethal genes and promoters exist. Novel synthetic constructs can also be used, opening up endless combinations and management possibilities. This debate is specific to dengue virus. However, because lethal gene technology prevents transmission by killing the mosquito vector, it can be used to combat all Aedes aegypti vectored pathogens. This technology has allowed the same lethal strain to be introgressed into local mosquito backgrounds, and these mosquitoes have been approved for release in countries throughout the world. This shows adaptability to different environmental and genetic situations. Responsibility is essential in any emerging technology. Lethal genes were built on the foundations of, and as an enhancement to, the sterile insect technique. Since 2009, lethal gene mosquitoes have been released, resulting in 80 to 95% reductions in local populations. For each country, these mosquitoes have gone through a thorough regulatory process and have been approved for release. Public engagement campaigns accompany each release, and a recent survey indicates Americans overwhelmingly support the use of lethal gene mosquitoes in the US. Importantly, researchers are testing for potentially unintended consequences. Modified mosquitoes have shown no increase in transmission of either dengue or chikungunya as compared to wild-type mosquitoes. Finally, lethal genes are simple to monitor. Modified mosquitoes are easily identified in the wild with UV light. Genetic sexing strains are being developed to pair with lethal genes, and this would cut rearing costs in half by only producing males, meaning our solution is more cost-effective overall. Now, while there are many positives for lethal gene technology, we feel it is also important to highlight why Wolbachia is the wrong choice. Essentially, Wolbachia is a black box. We don't know how or why it works, and its phylogeny is unclear, so it's simply defined as a species complex. Wolbachia do not naturally occur in Aedes aegypti. Scientists insert the entire foreign bacterial genome into the mosquito, which contains over 1,000 genes. Wolbachia is a form of gene drive that is not reversible. These mosquitoes are being released right now, and there's no way to recall them. Wolbachia organisms aren't versatile either. They can't be grown outside of their insect hosts, nor can they be engineered to improve their efficacy. Only 2 to 3 strains are known to reduce dengue transmission rates in Egypti. Wolbachia is negatively affected by a number of environmental factors. Mosquito eggs are often laid in shallow containers that can heat up quickly during the day. Exposure to these higher temps as immatures leads to adults with only a fraction of the Wolbachia density as those raised at lower temperatures. If the Wolbachia isn't there, it can't control dengue. Researchers are still unclear on how exactly Wolbachia affects the virus, and there is even evidence these bacteria can increase a mosquito's susceptibility to other pathogens. Even in release sites where Wolbachia supposedly has gone to fixation, scientists are not sure why they are now finding pockets of mosquitoes that don't carry the bacteria. There are further uncertainties for how Wolbachia interacts with the 4 different serotypes. In the words of Bull and Torelli, there are too many unknowns about such a process to make informed predictions, but the direction of evolution for dengue virus, Wolbachia, and the mosquito all coincide with intervention failure. So how can we ethically use this technology when so many unknowns still exist? The 2 technologies have the same end goal of decreasing dengue incidence, but they differ importantly in how to do so. Lethal genes kill the next generation of mosquitoes, eliminating the chance for transmission. Wolbachia only offers partial dengue blocking by an unknown mechanism. We've shown how lethal gene technology has been effective for mosquito population suppression in numerous countries for close to a decade. However, another way of using lethal genes doesn't suppress, but rather replaces mosquito populations with ones incapable of transmitting disease. This new method will essentially do what Wolbachia does, but better. Because it inserts known specific genes, there will be a recall mechanism, and ethical deliberations are currently being held in both the public and scientific communities about this developing technology. I think we all realize that one single technology will not solve the global health burden of dengue or any other pathogen. But for the purposes of this debate, there are many reasons why lethal gene technology is the best strategy to decrease the incidence of dengue fever worldwide. If we want to eliminate dengue, we need to eliminate transmission. Wolbachia are not up to that task. Lethal genes are. The choice is clear. Thank you.
Thank you, Team 1. And now Team 2 can cross-examine Team 1 for 3 minutes.
According to Massenet Burnell 2013, the laboratory experiments showed that 14% of the offsprings emerged from riddle males mated with the wild-type females. On a larger scale, 14% of the offspring surviving could be substantial. How could this problem be addressed?
In reference to you guys mentioning that a dead mosquito cannot transmit the virus.
So there has been 80 to 98% suppression of the local population. So yes, there is some survival, but still that decrease in the population is, with enough releases of transgenic mosquitoes, would be enough to eliminate transmission. Also, it's not necessary to get rid of every single mosquito. You just have to get below the transmission threshold and to eliminate transmission of the pathogen, but you don't have to eradicate all the mosquitoes.
But can a female be able to transmit the disease if they feed on an infected person?
Sorry, say that again?
I said, can a female vector the disease after feeding on an infected person?
Yes, a live female mosquito would vector.
No, the riddle, the transgenic mosquito with lethal genes.
If any transgenic mosquito didn't die, then yes, it's able to vector the disease.
Okay, thank you.
You mentioned that there was specific techniques used to target females so that male offspring would still be produced using lethal genes. However, the paper that you referenced, Etche et al. 2015, did state that it wasn't viable and it's not expected to work, and they had unsuccessful field trials in Mexico because of lowered mating
Yes, that's not the strain that's being used right now. It may be developed in the future, but right now what's just being used is the OX513A, which is the sterile males.
So for the time being, it's not the best strategy?
No,
females?
we did not say that the female-specific strain is the best strategy. We are talking about the releases that are currently ongoing, which is not that strain.
I think you're confusing the female-specific strain with a genetic sexing strain, which happens in the lab prior to release so that you only produce males.
So which strain would you guys say is the single best strategy?
The OX513A.
Using lethal genes to kill mosquitoes is the single best strategy.
You can use lethal genes to kill mosquitoes with a variety of different genetically engineered tools.
So the overall goal for you all is to manage the vector?
Yes.
Okay, thank you guys. We can now have Team 2 with their introduction presentation.
Good morning. Good morning. Biological control of Aedes aegypti using the endosymbiont bacteria Wolbachia is the best currently available strategy for decreasing dengue virus incidence worldwide. Wolbachia pipientis is a bacterial endosymbiont found naturally in over 60% of insect species, but is notably absent from Aedes aegypti, the primary vector of dengue fever. However, this bacterium can be artificially introduced into the vector. To do so, Wolbachia found in another insect is injected into the eggs or the abdomen of an adult Aedes aegypti. As the bacterium multiplies within the cells of the host, it also disperses throughout somatic and germline tissues. Of greatest interest is the phenomenon in which a mosquito transinfected with Wolbachia in the laboratory passes on fewer viral particles to the human host than non-infected individuals. In fact, replication of viral particles is reduced such that these mosquitoes can transmit as little as 10% of the viral load that uninfected mosquitoes transmit. In this way, mosquitoes could remain in the environment, but with the reduced capacity to transmit disease. All of the different serotypes of dengue virus are blocked by Wolbachia infection, making this technique one of the most promising strategies for controlling dengue fever. Moreover, Wolbachia infection seems to also limit infection by other disease-causing agents, such as chikungunya, malaria, yellow fever, and Zika. Release of Wolbachia transinfected mosquitoes would have little impact if there were no way to ensure that the bacterial infection would become established in the population. However, Wolbachia is vertically transmitted by the female to all progeny and exhibits a feature known as cytoplasmic incompatibility, which prevents females without Wolbachia from producing viable offspring after copulation with Wolbachia-infected males. In this way, only matings between uninfected mosquitoes will result in uninfected offspring. All other pairings result in either non-viable embryos or Wolbachia-infected offspring. The rate of cytoplasmic incompatibility is greater than 99%, so if sufficient numbers of transinfected mosquitoes are released, the Wolbachia infection will quickly spread as the chance of uninfected mosquitoes mating with Wolbachia-infected mosquitoes increases. Eventually, virtually all females in a population will become infected and have a reduced capacity to transmit disease, but will still remain in the ecosystem. Several field trials have demonstrated that Wolbachia can quickly spread and become established in a population. In 2011, mosquitoes infected with WML strain of Wolbachia, taken from Drosophila melanogaster, were released in 2 populations near Cairns in northeastern Australia. Over the duration of 3 months, an average of about 15,000 mosquitoes were released weekly over each 1,100 hectare area. 5 weeks after the field trial ended, monitoring efforts revealed that Wolbachia infection reached almost total fixation in both populations. Even more promising, Wolbachia remained at near fixation levels even 2 years after releases of Wolbachia-infected mosquitoes had ceased. Thus, it is clear that Wolbachia is capable of spreading and becoming established in a natural population. Field studies in Vietnam and Brazil included releases of Wolbachia-infected eggs, pupae, and adult Aedes aegypti, and showed that the releases of early life stages may be more sustainable and cost-effective than releasing adults only, by reducing the amount of resources dedicated towards rearing adult mosquitoes. Researchers not only measured the rate of Wolbachia infection in the target mosquito populations, but also measured the capacity of these trans-infected mosquitoes to block dengue virus 1 year after the release. Wolbachia trans-infected mosquitoes in the wild maintained the capacity to block transmission of dengue at levels comparable to those observed in laboratory trials. Models suggest that Wolbachia can reduce dengue incidence by more than 80% and can decrease the duration of dengue outbreaks by up to 6 weeks each year. How likely dengue virus is to escape viral blocking to Wolbachia remains uncertain. Theoretical models posit that viral escape from blocking should be highly selected for, resulting in a decrease in virus blocking in less than a decade. However, populations of mosquitoes that have been trans-infected with Wolbachia maintained viral blocking even after 2 years in the field. If the virus were to escape viral blocking, mosquitoes with multiple strains of Wolbachia could be released to curtail resistance. These super-infected mosquitoes have been shown to have equal fecundity and capacity to reduce viral transmission as singly infected mosquitoes, and they exhibit cytoplasmic incompatibility with mosquitoes that have only one strain. In this way, the super-infected mosquitoes would become established in the population and limit the likelihood of viral escape from blocking. Using Wolbachia to limit the transmission of dengue has several advantages over other strategies. First, it is cost-effective. Multiple releases of mosquitoes over just a few months could be enough to bring Wolbachia infection into near fixation levels, release of far fewer mosquitoes than are required for other strategies such as genetic modification. Second, it is self-sustainable. After just a few releases, the biological control agent naturally proliferates throughout the population, requiring no further supplementation. Third, Wolbachia is incapable of horizontal transmission to humans and other animals, making it safe. Fourth, it satisfies current regulatory standards and is already approved for use in at least 7 countries. And finally, it is safe for the environment, as it is a method for controlling mosquitoes and dengue virus without eliminating or eradicating a species, which could have unanticipated consequences for numerous animals that rely on mosquitoes as food. Furthermore, eliminating one species of mosquito may open up the niche for another species to exploit, which may result in the introduction of a new vector. If this were to occur, elimination of the new species would be required, including investment in developing new lines of genetically modified mosquitoes. Thus, eradicating species of mosquitoes to control arboviruses, such as dengue virus, is not an ideal strategy. Arthropod vector diseases are on the rise across a globe that is increasingly connected, and the insects and viruses know no borders. As a result, we must be united in our fight against arboviruses, striving to work together with the best possible tools to protect human health worldwide. Using Wolbachia as a biological control agent is the safest, most cost-effective, and most assured strategy to manage the global spread of dengue fever. Thank you.
Thank you very much. And now we can move into the cross-examination from Team 1 to Team 2.
Your sources, Yi, 2015, Ferguson, 2015, Frentiu, 2014, and beyond, all tested Wolbachia blocking properties at 25 to 27 degrees Celsius. How would pathogen blocking properties of Wolbachia be affected in release areas with temperatures above 30 degrees Celsius or 87 degrees Fahrenheit?
That's a good question. The main evidence for the blocking being maintained is that even in natural populations in Australia where it's hot, they maintain viral blocking even for 2 years after the release had ended. So those are all infected mosquitoes that had passed on to future generations and maintained viral blocking even those hot temperatures.
Why is there no Wolbachia blocking effects seen in mosquitoes that have naturally occurring infections such as Aedes albopictus and Aedes fluviatilis?
We're citing beyond 2010.
Why is there no Wolbachia blocking effects seen in other Aedes species that have naturally occurring Wolbachia, such as Aedes albopictus and Aedes fluviatilis? That's from beyond 2010.
Aedes albopictus actually passes on fewer viral particles because of Wolbachia present. They do not pass on as much as uninfected Aedes aegypti.
What is the molecular mechanism of WML, of virus blocking?
The infection of Wolbachia leads to the rise of oxidative stress and an increased level of reactive oxygen species in its host. The reactive oxygen elevates linked to the activation of the toll pathway. This immune pathway is responsible for activation of the antimicrobial peptides, which includes defenses and sarcopins. These antimicrobials has been involved to inhibit the proliferation of dengue.
Is that a theory, or is that proven
No, that's
as a mechanism?
Reference on PAN to et al. 2011.
Yeah, but is that still just a hypothesis?
No.
No. How do you propose to use Wolbachia as a reversible method of control?
Wolbachia isn't reversible, and that's why we think it's the best strategy, because in poor countries where dengue is endemic, they don't have the funds or the resources to continually release mosquitoes for control purposes. So our method, you'll do it once and it'll work, and they won't have to spend money again.
So how far has the release area in North Queensland spread over this 2-year period, saying you only need to do it one time?
Okay, you guys can continue because you have your first rebuttal period of 3 minutes, so. Okay.
There's very minimal spread of this throughout those areas because they don't travel as far, and so where you release, it stays, and the spread is not as much.
In your introduction, you stated that lethal genes could be used to replace mosquito populations. However, the two papers that you cited, Esvel and Akbari, both support potential strategies that are not currently being used. So how can this be accomplished now as the single best strategy? Or was it just a potential?
In House of Lords 2015, the committee mentions that only population suppression strategies and not population replacement strategies would be easily commercializable. And this is partly because businesses who would rely on the funds, they would release it once and then they would no longer be able to have income. But with these population suppression, they could have a continual release. Could there be a conflict of interest between those selling riddle technology and those relying on it who may be unable to afford such technologies?
Your source, Gorman et al., 2015. When surveying for Aedes albopictus abundance after riddle releases had suppressed Aedes aegypti populations, the author concluded that Aedes albopictus does not fill the niche. However, this trial was really short term, and the monitoring had stopped less than 2 weeks after the last release of those mosquitoes. Do you see any problems with this trial being cut short?
We also recognize in several references Oxitec 2016, LaRoche 2012, and Harris 2012. Wild types Aedes aegypti has addressed, genetically modified Aedes aegypti have short-term lifespan up to 4 days. Can riddle mosquitoes effectively compete with wild type adults that survive 2 to 4 weeks?
So models predicted approximately 3,150 males per hectare would be required for suppression, according to Harris et al. 2012. However, realistically, only about half that many, 1,400, were released per hectare over a 55-hectare area. This area was subsequently reduced to 32 hectares and then 16 hectares. With production difficulties mandating a continual decrease in the size of the area tested, what are the implications of the scalability of this technology to larger areas?
The supplementary material to Harris et al. 2012 cites that riddle is best used in an integrated approach alongside cultural control methods to reduce native mosquito abundance. Can riddle truly be considered the single best strategy if it's reliant on other methods for success?
And as discussed in Resnick 2014, amphibians, bats, birds, and other species feed on mosquitoes at various stages of their life cycle. What would the consequences of removing these species from the ecosystem be?
Thank you. And now Team 1 can enter their first rebuttal period of 3 minutes.
I just wanted to address the paper that you were referring to where they had to scale down the release size. That paper was done in 2010. The release sites and the scale have been scaled up since then. If you look at the Brooks 2016 paper, there's a new facility that should be able to reduce the impact for people. Up to 300,000 people should be able to release mosquitoes on that scale. So yes, they had to scale down, but the limitations were the rearing facility and not the technology itself.
You also asked about us citing gene drive papers, the Kistler and Esfelt papers. While there's only been one effector gene made so far, that is a strong gene drive strategy for us. So we still want to be thinking about that so then we can address all the questions, all the ethical questions and everything. And then we also have it in our back pocket for when more effector genes are made. So yes, it is still potential and we haven't done it yet.
But it's important to think about these in a responsible manner before releasing a gene drive system.
And you mentioned riddle as being the only strategy we're mentioning. Lethal gene technology is a suite of strategies, and it's not just the one that Oxitec is releasing. So we have a lot of management possibilities through the use of different lethal genes and promoters.
Also, we wanted to address, you said that the WML dengue blocking has to do with the reactive oxygen species and immune system boosting, but the conclusion in that paper was that the mechanism, there's still really a lot to understand, and that you have to understand that that blocking mechanism may be different for each different Wolbachia species. So there's really complex interactions between the pathogen and the mosquito, the pathogen and the different strain of Wolbachia, as well as the mosquito and the virus. So even if there's a little bit of understanding about WML and that interaction, all the other different actions are unknown, and it's unknown how the virus, the mosquito, and the Wolbachia are going to co-evolve together over time.
Z 2015 Beyond 2010 suggests using a single cytoplasmic incompatibility gene from Wolbachia instead of the entire genome. This would not be a feasible strategy when Wolbachia can't be cultured in the lab. In addition, Jobert 2016 suggests that a superinfection with 2 Wolbachia species to replace WML. However, the superinfected strain would likely not spread over the already established line because these eggs hatch later and in fewer numbers.
And that superinfection paper suggests using MEL and ALB. Well, ALB has been established in Aedes aegypti for even a longer time than MEL. It's been established for over 10 years, but this is not being used in releases. So if it's a good strategy, why is it not being used in releases? Why is only WML being used?
Thank you. And Team 2, you can go into your second rebuttal of 3 minutes.
In response to the Wolbachia and the superinfection, superinfected mosquitoes, while an excellent strategy, may not even be necessary. Were we to release only male-infected Wolbachia, we could knock down the population, then subsequently release female-infected mosquitoes, and then replace them with the new strain of Wolbachia or the superinfected strain of Wolbachia.
So Master ETR 2016, ratios as high as 50 to 100 oxy 513A males to 1 wild male are foreseen to be necessary to suppress the wild populations of Aedes aegypti. How are you going to maintain the releases in developing countries when you said it's hard to have these facilities in these developing countries?
I want to respond to your question about coevolution and the uncertainty of what will happen because we have different genes from virus and bacteria and mosquito interacting. And this is a very good point, and it highlights one of the most challenging aspects of Wolbachia. However, one of the biggest advantages is that there is evidence now that it actually works, that 60 to 90% of viral particles are blocked. And if we have such a powerful tool right now, it makes sense to use it at least while it lasts. As we said, there's projections that it may not last more than a decade, but again, it could go the other way and last even longer than we project. So it's worth using.
In response to your response about our Harris question, you said that the limiting factor in the Grand Cayman Island studies was the size and the capabilities of the rearing facilities. Again, in poor countries, will they be able to build and maintain these sorts of facilities?
Okay. Also, when you cease releases in the Cayman Islands, the main goal was to manage the vector, but was there a rebound after you cease releases?
So your reference, Resnick 2014, mentioned the Cartagena Protocol on Biosafety, and it said it requires participating nations to develop laws pertaining to safe handling and use of genetically modified organisms to protect human health and biodiversity. Well, first off, you're reducing biodiversity by eliminating a species, but second off, there's over 160 member nations of the Cartagena Protocol. How would you strike a balance between safety and precaution, all these different cultures, and the ethical responsibility to fight the disease?
Thank you. And now for your second rebuttal period.
In response to the cost of the rearing facilities, while there may be a need to release more mosquitoes initially, if you suppress a population, you don't need to do as much post-surveillance as you would with Wolbachia. With Wolbachia, there would be a lot of long-term costs after. You need to make sure that the mosquito and the Wolbachia are still blocking the pathogen. You constantly have to do that surveillance. You can't stop because you don't know when evolution is going to occur. Those post-surveillance is very difficult and would be very costly. Additionally, I don't know why Wolbachia rearing facilities would be any cheaper than our technique.
And also, recently a rearing facility was created in Mexico, which some countries considered a developing country.
You also mentioned that we are trying to eradicate the species, but we're not. We're trying to reduce the population low enough that the disease spread that dengue no longer is transmitted between people. Once the releases of mosquitoes are stopped, the population will rebound. That's part of the strategy so that we don't have long-term ecosystem effects. But furthermore, even if we did eradicate Aedes aegypti, it's an invasive species where dengue is a problem, so the concerns about niche change over time are less because it is typically an invasive species where dengue is a problem.
Also, you mentioned the evolution. So resistance is a problem for both strategies, but for lethal genes, there's only one possible avenue of resistance, which is through the evolution of the mosquito, whereas the Wolbachia strategy has three possible avenues of evolution of resistance, which is the evolution of the mosquito and the evolution of Wolbachia, and also the evolution of all four dengue serotypes.
And you also mentioned that the idea is to use this as an irreversible technology, but many of the papers cite that this is a big concern. Using something, you don't have a backup if resistance is to emerge. The superinfection, you don't know that they necessarily work, and although there are a lot of potentially available Wolbachia strains, it's irresponsible to release something right now that you can't take back from the environment.
Also, going back to cost, we're only releasing males, and we don't have to release males and females, which is, for us, is more efficient. So, and you guys will have to release both males and females, which will increase rearing costs, because you'll have to feed both males and females.
Thank you to both teams. We will now enter into a 10-minute question and answering session. We will allow questions from the judges first before the audience, but if audience members do have questions and there are time for your questions, there is a microphone that is on this side of the stage on the floor, so if you could line up next to the microphone with any questions that you have, that would be great. I also want to advise both teams that if a judge or an audience member has a question to both teams, just allow time for the other team to answer to be fair. So first, do our judges have any questions?
All right, we're on. So pretend like you're now no longer debating and you guys are working collaboratively to do some kind of release. In what way do you see perhaps Wolbachia and lethal genes being implemented at the same time in the same area? Is this possible?
So actually, that's a great question because two of the original Wolbachia papers suggested using Wolbachia as a gene drive mechanism to introduce transgenes, antipathogen transgenes, into populations. It wasn't until later that they discovered that it has dengue blocking properties. So gene drives could be used to, Wolbachia could be used to introduce genetic engineering technologies into the species.
And if used in this method, it would no longer fall under biological control. It would then be a genetic engineering technique. Also, you could use lethal genes as a population suppression approach, and then you could use Wolbachia as a population replacement, theoretically. So in that way, they could work together.
Well, we were excited for the first answer, but the second answer is exactly what we were going to say. You could knock down with the population suppression, and then follow up with replacement with Wolbachia, and it'll be more efficient. Far fewer mosquitoes will be required to be released when the population is already knocked to low levels. But yeah, because the cytoplasmic incompatibility already does that for Wolbachia, so we could just release males that have Wolbachia, and it would do the same thing as genetically modified organisms would with the lethal genes.
Also, if the mechanism was really isolated, we could use a lethal gene to spread an effector gene that we got from Wolbachia if those genes were isolated and identified.
Okay, does our other judge have a question? No questions? Are there any audience member questions? Could you please, come down to the microphone? Thank you.
Thanks. So my question is to Wolbachia team. So with regard to the culturing and distribution of Wolbachia, is there an easy way to do that? And is that a reason that the transgenic varieties might be better, given that Wolbachia is hard to work with?
So the initial introduction of Wolbachia into Aedes aegypti took about 4 years. It was very difficult to do. However, as soon as the laboratory populations were established, because maternal transmission is 100% to offspring, there's already colonies with Wolbachia, so we do not have to create new strains.
So in that case, it's got a very easy setup time. It's got a very long setup time, so it'd be very hard to use another Wolbachia strain. Would that be correct?
That's a good point. Subsequent introductions of, for example, Alb Wolbachia strain from Aedes albopictus into Aedes aegypti has taken far fewer than 4 years. So after the mechanism or the strategy was discovered, it is reduced time for introduction of Wolbachia to the mosquitoes.
And is that because we've learned how to do it better?
Exactly.
Okay, thank you very much.
Thank you. If there are any more audience questions, they can go. Please walk down to the mic. Thanks.
I was hoping that each team could take a minute and just talk about how they would attack the social or cultural challenges to implementing their strategies where they want to do these releases.
So genetic engineering has been criticized in the past for having poor transparency or poor public engagement. However, efforts really have improved, and especially in the releases in Brazil, where there is a radio jingle used, and there are festivals where people dressed up as mosquitoes, and there are education for children in schools, and different surveys and community meetings. So you really have to tailor the engagement to the specific area where you are releasing, which would be different around the world depending on the culture. So you do something very different in the United States than you might do in Brazil, or other places where you want to release?
So according to McNaughton and Duong 2014, they were able to design a community engagement framework for dengue fever. So they use social research tailored to the concerns, expectations, and socio-political setting of local, national, international, local, international, national setting they were working in. So they use radio, advertisements, posters, flyers, they had local awareness events, workshops, and visits to the homes of residents to spread public awareness and release sites in Australia, Vietnam, and Brazil.
Also, there's a high public aspect involved with Wolbachia. Because Aedes aegypti are very commonly associated with human dwellings, we have the ability to give the public a container with Wolbachia-infected eggs, and the mosquitoes are now in the area where they're most needed. So the public is involved very highly in that.
Okay. Thank you, guys. One more question?
Okay, this is a question for both teams. Let's imagine that both strategies work perfectly, as well as you'd hoped or better, okay? So with Wolbachia, you can convert the population so all Wolbachia are infected, and that probably better than currently completely prevents dengue transmission, okay? And with the lethal mosquitoes, you can eradicate the population in the target area, so in the areas that you want to treat it, okay? So you've got two different outcomes. One is converting the population to a non-dengue transmitting form. The other one is eliminating that population. Which would you prefer and why? And you might like to contrast where the mosquito is invasive, so in the Americas and Southeast Asia, versus where it's native, which would be Africa, or you might not.
So even if you have transformed an entire population through population replacement, there is still a concern for coevolution over time. And if we have completely suppressed a population of mosquitoes in the target area, there is still some concern for reentry or migration of mosquitoes from the outside area, but mosquitoes don't really travel very far distance, maybe 50 to 100 meters in their lifetime. So although it's something that needs to be addressed, not as much of a concern as having a population that is transformed and doesn't transmit dengue, but over time that may completely reverse.
So we would continue to support a replacement strategy because the elimination of something from an ecosystem of a species, the consequences of that are unknown and they're irreversible. And so we don't want to make any choices that could permanently change the ecosystem in harmful ways. We will not be removing things from the ecosystem, we will be changing what's already there.
I'd like to follow that up with the release of Wolbachia, of course, is also irreversible. However, Wolbachia exists naturally in over 60% of insects and has yet to be implicated in any kind of human diseases. And so the fact that it would be irreversible poses very little risk factors.
But dengue, Aedes aegypti, and we're talking about Aedes aegypti, has no naturally occurring Wolbachia infections.
Right.
And something else that you have to consider too is that a lot of countries are pretty strict on genetic modification laws. So because Wolbachia is a biological control, it'll be easier to get past a lot of those regulatory thresholds and hurdles that you would need to quickly and effectively implement in certain areas.
Actually, I think Paul Jabera from CSIRO stated that, I don't know if you saw his talk, but that for genetically modified mosquitoes and in the US and for Wolbachia in Australia, they actually went through a very similar regulatory process.
They're both regulated as veterinary drugs, both in Wolbachia in Australia and then lethal genes here.
Yep.
So the prompt for this debate was the single best strategy to eliminate dengue worldwide. And so in this hypothetical situation where both strategies work perfectly, we believe that Wolbachia will be easier to do and will be cheaper.
It won't be cheaper because you have to continuously surveil for evolution, and then you'll have to develop new strains and find new strains of Wolbachia that also block dengue that you can introduce into Aedes to replace the ones that have developed resistance.
So monitoring would still be necessary with GM mosquitoes because you have up to 14% of these mosquitoes emerging that can transmit the disease. So you would still need to be doing monitoring either way.
Yes, but monitoring with the UV light's a lot cheaper than monitoring with PCR reactions.
hospital records would be our monitor of decreased dengue.
Okay, that was it. So if we could please thank both of our teams.
Me too. So scared.
We will now have a 10-minute break until our third debate of the morning. Okay, good morning, everyone. If we could go ahead and get started with the third and last debate of this morning. So we have Auburn University and we have ICI PE this morning, and we want to first thank both teams for being so flexible with some of the challenges that we faced this morning, so thank you both. And we will start off with our unbiased introduction. Okay.
So thank you everyone for coming out this morning. My name's Edmund Norris from Iowa State University, and I will be doing the unbiased introduction for the third topic, the most important invasive arthropod species internationally. The biological diversity present in many stable ecosystems evolved over thousands or millions of years, producing a complex system of species interdependence and interrelationship. These diverse and natural ecosystems are generally capable of multiple biotic and abiotic insults, should they arise. Invasive species, however, can cause drastic changes in these ecosystems, leading to the complete eradication of various keystone predators or autotroph species. As such, many invasive pests can leave a lasting effect on their newly founded environment, and if the insult is sufficient, these invasive species can cause massive changes in biodiversity and significant economic loss. The introduction of various invasive pest species happens by a variety of means, and unfortunately, these events are occurring more frequently in our rapidly expanding global economy. These introductions may be intentional or accidental in their nature. Among intentional releases, motives have ranged from highly romantic, such as the introduction of starlings in 1890, simply due to their mention in a variety of Shakespearean plays, to practical, such as the release of the cane toad to control cane beetle populations in Australia. However, accidental introductions are far more common and potentially far more damaging. Although our global economy has led to speedy transport and an abundance of commodities, global trade routes and mass human migration has facilitated the spread of these invasive species in ways that were previously unimaginable. The spread of the Asian tiger mosquito, a competent vector of a variety of quickly spreading arboviruses, is a perfect example of a pest that is currently carried to new environments by means of the global economy. By hiding in small containers and tires, this pest can be trafficked to a practically unlimited number of environments. This example should serve as a cautionary tale in the transport of goods throughout the world, as these invasive species can lead to enormous damages. The effects of invasive species can be indirect, in that they harm species or aspects of the environment that other economic or ecologically significant organisms rely upon, or direct, which can lead to the destruction of commodities. Indirect effects are diverse and sometimes difficult to accurately measure or describe. Specific examples of entomologically relevant indirect effects include the ecological insult brought about by the destruction of plant roots by the European cranefly, and the varroa mite, which is capable of decimating commercial honeybee colonies used for the production of honey and pollinating a wide variety of commodity crops. Direct effects are exemplified by organisms that cause the immediate destruction of commodities or are significant vectors of medical or veterinary disease agents. Specific examples include invasive Culicoides midges that have been able to vector blue tongue virus to livestock, and the brown planthopper, which is capable of destroying rice fields that supply a very large percentage of the world's food supply. Moreover, problems associated with invasive pests can be characterized in multiple ways and can range from matters that pertain to public health, agriculture, forestry, aesthetic depreciation of commodities and property, to the destruction of natural ecosystems. The variety of damages associated with pest species makes their control paramount, but can also cause the implementation of control strategies to be quite difficult. There is no single method which can prevent the damages associated with all invasive pest species. Instead, control strategies must be employed which rely on an advanced knowledge of the pest's biology, its abundance, and the scope of its impact on natural ecosystems. As such, we need to be prepared to deal with these invasive threats quickly. By identifying their presence in various environments or agricultural settings, characterizing their biology, and implementing targeted and integrated pest management strategies, we can limit their long-lasting effects on our environments and our economy. For this debate, the teams have been tasked to describe the economic and ecological impacts of a chosen pest. The debating teams will then demonstrate the need for immediate appropriations from an unnamed, well-endowed funding agency to combat the threat via a variety of control techniques. Because the impacts of invasive pest species are so diverse, ranging from economic loss to matters of public health, teams will need to make their case and convince this agency that the threat posed by their respective pest species is more significant than the other teams. The pests in question are the varroa mite, represented by the International Center of Insect Physiology and Ecology, and the brown planthopper, represented by Auburn University. The winning team will adequately demonstrate the importance of the particular pest and secure funding from the hypothetical funding agency. May the most pestilent pest win.
Thank you very much. We will now begin with Team 1's introduction presentation.
I'd like to quickly acknowledge the absent students who did all the hard work and preparation for this debate.
So on behalf of Team ISIPE, we are going to be here to try and convince you why it's important for us to fund research on varroa mite, varroa detractor as an invasive species.
So Albert Einstein said that mankind will not survive more than 4 years if there was the extinction of the honeybee. So this might seem a bit of an exaggeration, but because many of the staple foods, such as wheat and rice, corn, potatoes, are not pollinated by bees. However, many of the fruit and vegetables that we do consume, which is about 35% of our diet, are.
And therefore, honeybee are very important. They can increase crop production by up to 96%. Worldwide. Europe and America particularly are known to depend heavily on migratory bee pollination, and insect pollination alone contributed to 9.5% of the world agricultural output in 2005. This translates to, out of this, $38 billion was as a result of honeybee pollination. Insect pollinators pollinate about 80% of most wild plant species, and these are very important in terms of the biodiversity. And alternative income sources that come from honeybee include honey, wax, propolis, as well as the royal jelly, and these are important sources of income.
So at the moment, there's a rapid decline in the European bee populations across the world. So in North America and Europe, there's been a loss of 30% of their managed colonies every winter. And so the number of beehives have only increased by about 45% since 1961, and this increase is not meeting the demand of pollination that is needed around the world. So this is a major threat to food security and biodiversity.
There are a number of factors that are resulting in the decline in honeybee colony that we observe in the world today. Some of them include the climate change, which, as we all know, affect all aspects of the ecosystem. Also, habitat alteration. Somebody was talking earlier on about the changing the plant diversity. But more importantly for the debate today are pests, pathogens, and diseases.
So varroa destructor is currently recognised as the most important invasive pest of honeybees, and it contributes to the worldwide phenomenon known as colony collapse. And so direct effects include physical deformities, such as reduced wing virus, vitality and longevity. And indirect effects is that the varroa destructor does pass on viruses that leads to the deformed wing phenotype seen in some bee colonies and many other viruses.
and has colonized all continents except Australia. Currently, Australia has very good border control policies, so we've kept the pest out so far.
Several methods have been used previously in trying to manage this mite. These include chemical application, use of synthetics, pesticides, natural products, acaricides, and organic acids. Also, biotechnology has been employed in controlling the varroa mite, which entails the drone brood removal. Also, we've had bee breeding practices as one way of controlling the varroa mite. And overall, these have not seen the desirable results. And this is the reason why we are advocating for more funding to investigate further and be able to come up with new control tools that can be used to control these pests.
So an example of how colony collapse can affect individuals. So in Madagascar, a retired primary school teacher turned to honeybee farming in 2010 as income support. And they had 20 colonies, which was harvesting 120 kilograms of honey a year. And so the value of honey is 5 kilograms, so they were bringing in $12,000 worth of income. However, when the varroa destructor mite invaded their bee colonies, the colony was reduced by 75%, and so unfortunately the primary school teacher had to go back to the classroom because they couldn't support themselves due to their collapse of their hives.
Now, I want to convince my opponents to cross over and come and help us to advocate for the research on varroa mite. There is an excellent center which has been set up in Africa, in ISIPE, which is the African Bee Reference Laboratory, which is trying to answer a number of questions. One of the key factors which have been found is that the African honeybee is resistant to varroa mite, and it has been hypothesized that it uses a number of mechanisms to be able to resist this mite. And therefore, ISIPE has set up this lab to try and answer some of these questions. Is it true that African honeybees, the subspecies, are resistant to varroa mite? And if it's true, how do they do this?
So our take-home message is that we need to invest in the control and containment of a varroa destructor. Otherwise, the human population could be destroyed within 4 years if we lose our honeybees.
Thank you for the attention.
Thank you very much. Team 2 will now have a 3-minute opportunity to cross-examine Team 1.
So in your presentation, you mentioned that varroa honeybees pollinate some wild species. Well, my question is, what are the staple crops that are utilized worldwide that are dependent on human survival that have been pollinated by honeybees?
Okay, we gave a citation there that up to about 35% of the world food production is dependent on honeybee population. Well, honeybee pollination. And out of this 35%, there are the fruits and vegetables. And if you go to the market, for instance, to ask for a kg of rice, it's about $3. A kg of apples is $4. So in terms of quantity, it might look like it's a small contribution, but when you look in terms of value, then it's very significant.
Now, did any of your references deal with habitat loss? And if so, how does habitat loss impact bee decline?
So habitat would affect bee loss because bees need a variety of pollen to keep themselves healthy and keep the hive going. So to build up their innate immunity to the varroa mites. So with habitat destruction of native flora that rely on honeybee pollination, that would inevitably affect the health of the colonies of the bees.
And there is a recognition of pesticides contributing to bee declines worldwide. So can you explain how your plan will reduce bee exposure to insecticides and fungicides? Because from your plan, you said it's multiple sources. So can you explain that?
Actually, pesticides and insecticides, they have been used, particularly in trying to control the varroa mite. They use miticides, and what we are advocating for are ecologically viable techniques. For instance, if the African honeybee is able to resist the mite, and if we can elucidate which mechanism they use to resist the mite, one of them being the grooming behavior and the hygienic behavior, can we then breed bee species which are able to control the mite without necessarily having to apply the pesticides?
There's a global decline in managed hives. How does your plan address increasing managed hives worldwide?
Okay, thank you. And now we can have Team 2 with their introduction presentation.
Thank you for your consideration, and we hope you choose to fund a proposal for brown planthopper research. We are seeking funds to connect researchers within Asia, as well as to provide support to rice producers. Asia houses nearly 1/3 of the world's population and is heavily dependent on rice as it has significant cultural, economic, and nutritional importance. Unfortunately, rice production has been outpaced by demand since 2000, with many countries remaining food insecure. The problem of global food security is exacerbated by continued population growth coupled with declining yields, land, and water resources. The brown planthopper, or BPH, is the greatest threat to rice production and global food security. Losses from BPH are often catastrophic, with annual losses from feeding and virus transmission exceeding 1 billion kilograms in China alone, with 2.8 billion kilograms lost in China and Vietnam in 2005 and 2008. This is an international crisis that must be addressed with a coordinated international response. You might be asking yourself, why rice? Well, what else is there? Asia produces and consumes 90% of the world's rice, and 53% of the world's population is highly dependent on rice for daily survival. It is the main source of calories and protein. Rice's global importance is further strengthened as rice provides an income for over 100 million households in Asia, Africa, and Latin America. In southern and southeastern Asia, 20% of the population is undernourished, and over 500 million people live on less than $1.25 US a day. Unfortunately, the adoption of continuous rice cultivation altered and augmented pest pressures, most notably by shifting the brown planthopper from an occasional pest to a constant threat. Rice is the largest food staple in the world and is not dependent on insect pollination for production, and is the most effective way to combat undernutrition and poverty, not only in this region, but in Africa and Latin America as well. With an ever-growing world population, there's the need to increase food production for global food production for security, especially in developing countries. The brown planthopper became the greatest threat to rice production after changes in agricultural practices from the Green Revolution. The over-reliance on chemical insecticides and fertilizers further magnified problems caused by BPH, as many producers lack education on the pest biology and resistance management. The BPH is mellifera on rice sap, and its winged form is capable of long-distance migrations through Asia, through the Indochina Peninsula. Threats from BPH are compounded by its short life cycle, ability to vector diseases, and broad insecticide resistance to many chemical classes. Currently, Asia is the most food insecure region and will be greatly impacted by climate change with reduced yields. Within this region, food security means rice security. Annual temperature increases of just 1 degree centigrade can cause grain sterility, reducing yields up to 14% while still benefiting the brown planthopper. Increasing temperatures will drastically expand the brown plant hopper's overwintering area in Asia and result in additional generations, providing for larger reserve and immigrating populations. Additionally, rising sea levels, increasing soil salinity, and the loss of farmable lands will put more stress on an already fragile system. As mentioned before, the BPH is dimorphic with long-winged and short-winged forms. Wing morphism is influenced by environmental cues and can be a heritable trait. The long-winged morphs are capable of distance migrations, whereas the short-winged morphs are incapable of flight. However, there is a fitness cost for flight. The long-winged morph has reduced fecundity and a delayed start to oviposition compared to its short-winged counterpart. The selection of winged morphs with insect growth regulators has been met with limited success. However, all wing development genes were identified by Shui in 2015. RNA interference is able to disrupt wing development in both morphs. Building on that work, Lin in 2016 was able to demonstrate binary control of wing selection with RNA interference by the activation or suppression of 2 insulin receptors. Now that this pathway has been successfully determined, our goal is to incorporate this technology into new rice cultivars in overwintering areas that select for short-wing morphs until cultivars can be developed for field-level control. To effectively feed a growing world, we need to introduce new, high-yielding, environmentally friendly rice cultivars that have BPH resistance. Chen and Zha have already been able to confirm the compatibility of GM rice with systemic RNA activity against the brown plant hopper. However, they lacked appropriate target genes for further control, but this issue has since been addressed by Shui and Lin. With this funding, we aim to provide IPM education support to rice producers while connecting researchers. We plan to map and model migrating populations for efficient timing of control measures while developing new rice cultivars for BPH. By doing so, we can achieve food and economic security in Asia. Now that we've discussed the merits of our proposal, we'd like to briefly highlight some of the shortcomings of our opponent's proposal. Staple crops are needed to feed the world, and many are not dependent on honeybees or insect pollination for production. While knowledge gaps remain on the extent of pollinator decline, honeybees are not responsible for feeding the world. But what we do know is that a myriad of factors impact pollinator health, and that no specific cause has been identified as the sole reason for bee decline. We know that bee stressors do not act in isolation, and they often have a synergistic effect. Varroa is unable to survive outside of the genus Apis, and the heavy reliance on a few pollinator species runs the risk of a supply shortage. Additionally, yields have a stronger correlation with wild pollinator abundance than they do with honeybees. The brown plant hopper is the greatest threat to economic and food stability in Asia. Billions of people are reliant on rice for daily survival, with millions dependent on rice for economic security. Reduction in losses from BPH needs to come from a coordinated international response. Thank you, and we hope you choose to fund our proposal.
Thank you very much, Auburn. Team 1 will now have the opportunity to cross-examine Team 2 for 3 minutes.
You mentioned how important rice is to feed the world and provide many underprivileged people with their daily calories. Do you have a comparison of the calories of rice compared with other crop cereals?
No, I do not have a comparison of that, but somewhere around 80% or 70% of the world is dependent on rice for at least some of their daily calories, and over 53% of the population in the world gets a substantial, almost their entire daily calorie load through rice. So no other food or staple has any sort of daily caloric intake that even matches or comes close to rice.
Also, in addition to that, when you compare our target crop with the crops that are being pollinated by your bee, your insect, you find out that the calories and proteins that can be derived from our crop is far more important, is far more greater than the vegetables you guys mentioned that are being pollinated by honeybees.
Just a follow-up question. Do you mean to say that the world population is dependent on carbohydrates alone? And you mentioned about malnutrition. Is malnutrition only entails one nutrient diet, carbohydrates?
I didn't quite hear or understand. Could you rephrase or?
Okay, what I mean is when you are talking about vegetables and fruits, you're talking about balanced diet. And we all know that for a healthy living, you need a balanced diet. But when you're talking about rice, it's carbohydrate-rich food.
Ungunyen et al. 2002 from our presentation is stated that 53% of the world population are highly dependent on rice for daily calories and protein. So it's not only carbohydrates. Protein, which is very important for human growth and development, cell development, can be gotten from rice too. So not only carbohydrates. And carbohydrates is also important for energy. So it's very crucial.
I was wondering if you could elaborate on how host-specific the brown plant hopper is?
It only feeds on rice. And it's the #1 pest in Asia.
We have no further questions.
Okay, thank you. Team 2 can now enter their first rebuttal period of 3 minutes.
Or team 2?
Yes, you are team 2.
The consensus is that a major driving force behind long-term global pollinator decline is habitat loss. For example, during the 20th century, 97% of flower-rich grasslands were lost in the United Kingdom. This is partially a result of increased urbanization, which has resulted in bees shifting to monotonous diets. Potts et al. In 2010 deals with several of the issues dealing with bee declines. What is your plan for habitat and food restoration in an increasingly urban world? Also, we know that we want to really stress the importance of rice. Nearly 40% of the world's rice crop is lost annually to pests and diseases, and a lot of that comes from the brown plant hopper. There are 100 different pests of rice globally, but only really 20 that cause economic damage. BPH is the #1 pest, and it can vector 2 diseases, the ragged stunt and the ricey grass, which are also persistent and incurable. So when you get a virus like that, you kind of lose your crop.
Also, in addition, part of our proposal calls for farmer education and support, because you may be wondering that many parts of the world are not receptive to GM crops, and we're going to address this. But everyone is entitled to their own opinion, but not everyone is entitled to their own facts. So through scientific education, we hope to demonstrate the value and need for GM crops using the methods we highlighted from our proposal. And also, you may be wondering, how do we manage for GM rice? Will it work? And how also do we manage for crop resistance? We agree that more research is still needed to ensure that GM rice developed for this insect wing selection and eventual feed level control is effective. However, currently, several researchers, they are working on these issues, and they are finding and mapping several promising pathways. And the good news is that we are already able to successfully demonstrate the ability of GM rice to control BPH from feeding without any specific targets.
Additionally, we really want to educate farmers with IPM practices that they could have readily available, such as, using light traps to catch brown plant hoppers at time applications.
Thank you. And now, Team 1, you will have your first rebuttal period of 3 minutes.
So you mentioned the shortcoming of relying on a single or very few pollinators to pollinate many of the fruit and vegetable crops. but isn't a reliance on one particular food crop, such as rice, to feed 50% of the world population also a shortcoming? Also, it's very well and good of the research that is going into GM food, particularly in the GM rice, but a major issue is the public's perception of genetically modified food.
Okay, I also want to follow up on a question which you were asked earlier, that the brown leaf hopper is specific to rice. It has a very limited number of hosts. Is it not advisable then to think about rotational cropping? For instance, in Asia, where rice is heavily grown, we also know that other crop species are grown there, including wheat. So if leafhopper is specific to rice, if you do rotational cropping, then you are able to get rid of this pest, and therefore you don't really need to invest in further research to combat this problem.
Also, the honeybee issue is a worldwide issue because throughout the world, honeybees are pollinating fruit and vegetables, whereas rice is heavily relied upon in Asian countries, but we need to feed the whole world, not just the Asian countries. And shouldn't we be looking to steer away from monocultures such as rice and try to plant variability of our crops? So relying on wheat and corn, for example, which generally use a lot less water than rice.
Okay, thank you. And now, Team 2, you have 3 minutes for your second rebuttal period.
Okay. Quickly, the food reliance and saying that rice is, relying on one thing is dangerous, but I don't think that it is. And if you look at Asia, that consumes 90% of the rice, it has a long history of a cultural importance. It's really deeply rooted into these people, and to ask them to change their diets, I think, is not an appropriate way to go about it. It has, significant nutritional importance as well as cultural importance. It's used at weddings, it's given as presents, it's a sign of wealth. So I don't think we would need to steer away from monocots. You can augment landscapes around there by putting in floral resources that would provide nesting sites and habitats for bees. And again, rotation isn't really going to be a feasible option because they have to use continuous rice cultivation just to feed the population. So they constantly are planting and replanting rice. Say your honeybee issue is cosmopolitan. That's almost a double-edged sword. When you look at cosmopolitan and the Varroa relationships throughout the world, you really can see that based on the regions, the relationships and the factors affecting honeybee health are really dependent whether Varroa is there or not. And so,
also in addition, your first point mentioning the other percentage of the world which are not highly dependent on rice, how do we account for them? Remember this simple truth that food security in more than 50% of the world will surely negatively affect other parts of the world. As you can see the evidence in current migration in parts of Europe, and we have refugees in this part of the world and in the United States. So food security in Asia and in Africa, and we grow rice in a large concentration will surely lead to global food security. And talking about the public perception of GM rice, we already addressed that. People are entitled to their own opinion, their own opinion, but not to their facts. And a lot of research is being going on to also address this issue. And also, you mentioned about BPH specific to rice and not wheat. Remember these crops, rice, wheat, they are staple crops, unlike the food that is being pollinated by a honeybee. So staple crops has a more importance compared to the food that are pollinated by bees.
Tapping onto that, as a scientific community, we need to do a better job of connecting correct science information to people and policymakers. Simply can't feed the world without incorporating GM technology into this.
Thank you, Auburn. And now, Team 1, you have your 3-minute second rebuttal period.
Okay. I just want to bring to your attention here that when we are talking about the bee being important in terms of pollination, when you're talking about food, it's 35%, but in terms of the biodiversity, it's more than that, and it's always underestimated. And the world without biodiversity is definitely going to collapse, and the human population will collapse. So it's important for us to think about the biodiversity as we think about the food that we want to eat. And I think one of the driving forces that has been driving the climate change today is the human activity. So if we put more fields under rice cultivation, we are basically escalating the problems associated with human activity. And it's important for us to note here that the bee play a very important role in terms of maintaining this biodiversity. If we lose the bees, we lose the biodiversity.
So in your introduction, you mentioned a changing climate and a changing environment. So we all know that temperatures are increasing. However, a 2012 paper by Pierre Foncourt et al. Showed that brown plant hopper nymphs are already living at their maximum extreme. So increasing environment would be potentially beneficial to rice growers in decreasing the brown plant hopper populations. So therefore, more of our resources should be put into controlling the effects of varroa mite on the honeybee population.
I also want to pose a question here at this point, that which one is more important, to control a problem that is increasingly and escalating, like the collapse of a bee colony, vis-a-vis to control a problem which can be managed by alternative methods in terms of rotational cropping? Where do we need to invest our resources?
Also, coming back to the GM rice, I believe it's very hard to change people's perception, and that you'll need a really strong educational method to be able to convince people to use the GM rice, to change people's opinion.
All right, thank you both. Now we will move into a 10-minute question and answer session where we will have the judges ask questions first. Just as a aside to both teams. If a question is asked by one person to both of you, or two questions by one person, please keep in mind of the time to allow the other team to answer that question, so just to be fair for that. So we will ask our judges if they have any questions first. And for any audience members, if you have questions, there is a microphone on this side of the stage. If you could come up to that microphone, that would be great.
Okay, this question is for both teams, and I know you can't give a really lengthy answer, so just keep it very broad. So let's say you've got the grant, you've got a graduate student, and you're working with them to develop a project. Where would you start, and what's the broader impact? Because you do need to remember this is an international agency that you've pitched this grant to. So broader impacts, where do you start? Both teams.
Okay, that's a nice question. Regarding the timeline, where to start, we agree that it takes a tremendous amount of time and money to develop GM crops, as we are proposing for the GM rice. We would require additional industry support to develop new rice cultivars. And with almost half of the time that's required for this, it also includes regulatory and registration phases. Our hope is that the timeline could be sped up since the technology has already been discovered and developed. From our presentation, we showed some already discovered and developed methods. We just need to connect the information and the researches together, and hopefully this shouldn't take forever.
We'd really try and get those labs that we were mentioning before to connect and work together on collaborative works as well.
Okay, as we had indicated that we have a resistant variety, resistant honeybees, the African honeybee, and also some Russian honeybee, which have also been shown to be resistant to varroa mite. That is where I think we need to start. We need to understand what are the mechanisms that these honeybee varieties are using to be able to resist the varroa destructor. And then once we understand this, then how can we utilize the information that we have in terms of empowering the European honeybee, which is the most threatened group of honeybee. And when we understand this, it's going to impact the entire world, not just Europe, not just America, but I think even other African countries where perhaps this, the honeybee, the problem of the varroa mite has not been taken seriously. We can be able to understand how to.
Okay. Does our other judge have a question? No question? Is there an audience member with a question? Wonderful.
Hi, so my question's for the team B. So from my understanding, there's about 7 different honeybee species, and varroa disruptor only attacks 2 of them. Is that correct, first of all?
So yeah, varroa disruptor attacks the Asian honeybee subspecies and the European honeybee subspecies.
And so there's what, something like 44 other subspecies as well? Oh, I'm just going off Wikipedia, I was just reading earlier. So with so many other possibilities, does that not make it less of a priority? Can't we just change the cultivated species?
Well, we might say that is an option to cultivate the other species, but before you move into cultivating other species, you need to understand, can these other species adopt into the migratory kind of husbandry that is practiced for the European honeybee.
Okay, and the secondary one is, what about the role of commercially available bumblebees? Should there be a complete collapse of the honeybee industry? Couldn't every farm that needed pollination simply purchase their own hive from a commercial company, for example?
So bumblebees are known to push out native bee species. So there have been, I think, some studies done in Australia. So if you did use bumblebees to replace the European bee population, you could have a massive effect on the biodiversity of that particular area and lose native bees that are also important to sustaining biodiversity.
So it would be a problem, but it wouldn't be a human starvation problem.
Could potentially, if the specificity of the bumblebees couldn't pollinate the crops and the food that the European bumblebees currently do.
Okay, thank you.
Hi, I have a question for both teams. So in your prompt, your theoretical grants were submitted to an international funding agency, and because of increased trade liberalization facilitated by the World Trade Organization and globalization, agriculture increasingly exists in a global market. So can each team defend their stance in this context where food and pests are not only important for food security, but also for financial security through export of agricultural goods to other markets, other countries?
Yes, with rice, it's a little bit harder of doing that since many of those Asian countries are food insecure. A lot of those countries have actually put bans on rice export because they don't have those supplies and the reserves, if something were to happen. So unfortunately, we would like to see more cooperation within that region, but unfortunately, those countries feel that they have a stronger priority to feed their people first. But we would like to, if we could, help negotiate some rice coming out to help feed those people who are starving.
And also, in addition to that, rice generates income of more than 100 million households that are dependent on rice in the world. And like you said, though Asia, they may have some blockages to exporting, but they still do export to places like Africa, where I come from, so this is a fact I know, But so when they export, it's gonna serve as additional source of income to these parts of those who grow this rice.
Okay, we mentioned that besides pollination, the honeybees also have other products which are important. And in an effort to try and control the problem of varroa mite and other pests in this matter, there has been increased use of insecticides. Yeah, and if we understand how the African honeybee is able to, or rather the other honeybee subspecies are able to control the mite, then we might be able to come up with a controlled strategy which does not entail the pesticide. And obviously this is going to increase the export and the income.
Thank you. We have about 2 minutes left if there are any other audience questions.
I will say this, and maybe this will help, if we also look, it's been discussed heavily about public perception of GM and countries' receptivity to it. But if we look at this, more and more countries are realizing this. Currently, Argentina, Australia, Brazil, China, France, India, Indonesia, Italy, Iran, Japan, Mexico, Philippines, Spain, United States, Bangladesh, South Korea are all working on developing GM rice. So I don't know if anyone has felt that we didn't adequately address that.
I still think you're going to find issue with the public perception on GM food.
We agree on something.
And just to clarify, there has been one other host found for
Unless you're the cut rice, but it used to be a rhiza as well. It's just a weed that only occurs in rice.
You mean rice used to be a weed? Before it was cultivated.
Okay. Well, we can end right there. So if everyone would please give a round of applause to both teams. And that concludes the ICE International Student Debates for 2016. Thank you, everyone, very much.